Good afternoon, everybody, and welcome back to the Frontiers of Brain Health, where we take a deep dive into some of the most exciting and relevant science going on. I'm Dan Krosik. I'm the Deputy Director here at the Center, as well as a faculty member in Behavioral and Brain Sciences at UT Dallas. Just to remind people, we have a virtual audience. We will take questions both in person and virtually, so if you'd like from the virtual side to ask something, go ahead and use the Q&A function, and we will get to some of those questions at the end of this talk. Today we have a really amazing speaker and a really cutting-edge scientist, Dr. Emily Jacobs. Now, Emily is unique in a variety of ways. She began her career with a bachelor's at Smith College before being an instructor at Harvard Medical School. She then did her PhD at UC Berkeley in the lab of Mark Esposito, and so I actually overlapped a bit with Emily at that time. And she then went on to UC San Francisco, where she was a Robert Wood Johnson Scholar, and then is now an associate professor at UC Santa Barbara in the School of Psychological Brain Sciences, where she is currently working on her lab and developing young scholars from there. And she's gained a variety of recognition, which is very deserved. She's gotten a Brain and Behavior Young Investigator Award, a K-12 Career Development Award from the NIH. She also made it on the Frontiers of Science Distinguished Young Scientist Award or recognition from the National Academy of Sciences. She has a TED Talk on how gender diversity drives scientific discovery, very relevant topics, and her work is on a variety of important areas of neuroscience, including how hormones affect our psychological health, our cognition, and our emotions. She's done a lot of work on the menstrual cycle in women, and is looking at neurochemistry in a way that's very grounded in our psychological experience and how we live our lives. I can't wait to hear more about this. So let's go ahead and welcome Emily Jacobs.
Thank you for having me, not just once, but twice. It is a pleasure to be here. And if you were at yesterday's talk, that was really sort of the public-facing one. Today we're going to roll up our sleeves and get really nitty-gritty into the science. So bear with me. Okay, let's see if this clicker works. So if I ask you to close your eyes and imagine the human brain, you probably imagine something that looks a little bit like this. I always say it looks kind of like a 3-pound squishy walnut. If you were a neuroimager, maybe you imagine something that looks a little bit more like this. But as a neuroendocrinologist, we think about the brain a little bit more like this, right? Like in its full environment. And I like to say, John Morrison says sometimes that often scientists can be so taken by the complexities and the unique capabilities of the brain that we forget something really important about it, right? It's part of this larger integrated biological system. And our endocrine system is a perfect example of this sort of whole body integration. Oops. So in this talk, I'm going to focus specifically on gonadal hormones. These are hormones that are produced by the ovaries and the testes. They use the circulatory system as their superhighway, and they travel all throughout the body. And what's critical for this talk is that the brain is a target tissue for these steroid hormones, right? So the brain is an endocrine organ. We've got 20 years of animal studies that suggest that gonadal hormones, aka sex hormones, are powerful neuromodulators of learning and memory. Let's see if I can get the, whoops. I'm on screen, so I can just use my hand. So we know that there's this cartoon image represents sort of the interaction between our brain and our reproductive endocrine system. So there is this beautiful, tightly coordinated neuroendocrine cascade where your hypothalamus signals to the pituitary to then signal to your ovaries and your testes to start producing steroid hormones like estradiol, progesterone, and testosterone. These hormones then travel through your bloodstream and they bind to different target tissues wherever there's an appropriate receptor throughout the body. And as I alluded to, the brain is a target tissue for these steroid hormones. So there are regions of the frontal lobe and the medial temporal lobe tissues that express abundant populations of hormone receptors. We know that, for example, at least in non-human primates, that about 50% of the layer 3 pyramidal neurons in prefrontal cortex express estrogen receptors. This is a particular form called estrogen receptor alpha. And if you just look at sort of a correlational sense, if you look at individual differences between animals, those animals that have more estrogen receptor alpha in their prefrontal cortex show better performance on a delayed response task. This is work from John Morrison's lab from probably about 15 years ago now, but it gave us the first clue that sex hormones are not just responsible for guiding standard kind of reproductive behaviors, which is what most of the neuroendocrinology literature up until that point had really told us. These hormones are powerful neuromodulators of learning and memory. They're acting in extra hypothalamic sites to guide the cortex and to shape executive functions. So these kinds of careful studies have been conducted in animal models, but in humans we know a lot less. So over at UCSB, my lab is trying to get a toehold on how gonadal hormones shape the human brain. So here's some of the projects that we have going on in the lab. Rest assured, I'm not going to go through all of them. In fact, in this talk, I'm just going to highlight some of our precision imaging studies that map the brain across major neuroendocrine transitions. So I'll just focus on a couple projects here, focusing on the menstrual cycle and pregnancy. But I want to maybe begin, and if you're at yesterday's talk, this is a little bit of a repeat, but for those who weren't, I want to think about the fact that we're probably decades behind where we should be when it comes to women's brain health. As we wrote in a recent editorial, neuroscience has overlooked aspects of the human condition, the menstrual cycle, the pill, pregnancy, and menopause, that are relevant to half of the world's population and half of the US tax base, and we've got to correct course for the field to advance. Or in a more recent editorial, we say sort of the same thing, that women's health research has been historically undersupported. Now is the time to correct course. This is true also for the neuroscience of hormonal contraception, which we basically know nothing about. So if you want to ask yourself about the scientific body of knowledge, ask, whose body does it serve? In short, scientists can't answer questions they don't see. So we have a field in which 80% of tenured neuroscience professors are male. Maybe there's questions that have historically not been visible. So we need scientists whose vision isn't blurred by the lack of experience. And you don't have to take my word for it. The data sort of bear this out. So if there's any neuroimagers sort of listening in or in the audience, right, like you've all seen this stunning growth of brain imaging publications over the last 30 years. I call this the sort of blue wave. As a reference point in orange, you can see the number of publications using any kind of brain imaging tool in humans that have to do with depression. So that's the orange line. If you squint really, really hard, you can see the thin black line at the bottom. Right, that is the totality of articles covering menopause, pregnancy, hormone birth control, the menstrual cycle, and much, much more. So altogether, studies of these women's health factors constitute less than half of 1% of the brain imaging literature. By ignoring these factors, we risk making two major mistakes, right? First, the brain is an endocrine organ, which I hope I'll convince you of by the end of this talk. And yet we have almost no insight into how the dynamic changes in sex hormones shape the human brain at the mesoscopic and macroscopic scales afforded by human brain imaging. And second, we're missing critical clues about why these neuroendocrine transition states may be periods of both resiliency and vulnerability. So I'll just say that progress in neuroscience, I think, will flourish when the health of men and women are valued equally. Neuroscientists usually want to do one of three things, right? We want to understand how the brain works, we want to promote health, and we want to prevent disease. So let's talk about two of the leading causes of disability worldwide, depression and dementia. Here are two disorders in which women outnumber men 2 to 1. So of the 6.7 million Americans living with dementia today, 2/3 of them are women. Right? Similar pattern with depression, right? Women are at twice the lifetime relative risk relative to men. What happens, whoop, I got a little carried away, let me go back. So what happens if we take a step back and we think about these disorders through the lens of women's health over the life course? Right? You start to see a really interesting pattern emerge, which is that over the lifespan, pronounced changes in women's reproductive state are coincident with periods of increased risk for depression. Before puberty, we see rates of depression are similar in girls, but during the pubertal transition, you see this doubling of the rate of depression. Same thing with use of hormonal contraceptives, the perimenopausal period, and menopause itself. We know that depression can be a risk factor for dementia given the shared neural circuitry that gets disrupted in both disorders. And my group and others have found that, let's see, that earlier age at menopause, whether we're talking about surgical menopause or spontaneous menopause, that earlier age at menopause is tied to an increased risk of dementia. Based on work from folks like Roberta Brenton, Michelle Mielke, Karen Frick, and others, we're beginning to think of menopause as a neurological transition state that impacts tissues susceptible to neurodegeneration. The question is, what are the neuroendocrine drivers? Well, one striking feature of the brain and of these regions of the brain that are regulated by sex hormones is that they overlap considerably with circuits that are dysregulated in both depression and in Alzheimer's disease. So tissues like the entorhinal cortex, the hippocampus, the prefrontal cortex, at the network level, the default mode. So a critical question is, what are the physiological properties that make these hubs so prone to damage? Right? And what properties of brain health can provide some resilience? So I'm going to take you through some of the science that are, we're trying to kind of piece together some of these threads of thinking about how and where and on what time scale these neuroendocrine signals might be driving different aspects of the brain from structure and function. But before I do that, you've got to know a little bit of neuroendocrinology. This is obviously a cartoon, but it illustrates a really fundamental point, which is that one central feature of the mammalian endocrine system is that steroid hormone production, right, the secretion of steroid hormones, ebb and flow over time. It does this on different time scales. We can think about the diurnal cycle from AM to PM. But here's another really famous cycle, which is the menstrual cycle. So you can see this kind of canonical rise in estrogen, which peaks around the periovulatory period and then starts to decline. And then later, in the second half of the cycle, you can see this increase in progesterone and a decline. And for most women, for most of our lives, these rhythms are as steady as the tides. These pulses are almost like a vital sign. They drive physiological changes throughout the body, including likely the brain. And here's some of my favorite work. This is foundational work conducted way back in the 1990s by Catherine Woolley. And she was one of the first to link changes in hippocampal spine density to a rodent's estrous cycle stage. And here's what she found. So this is looking at a CA1 neuron in the hippocampus. And she's zooming in and looking at the dendrite and these little protrusions on the dendrite called dendritic spines. And what she observes, if she looks at diestrus, which is the beginning of, so I should say rodents have an estrous cycle, which is kind of like a human menstrual cycle, only it's truncated, right? It occurs over a 4 to 5 day window. And when estrogen levels are low at the beginning of the cycle, you can see this sort of sparse spines. On the day of ovulation, when estradiol levels peak, spine proliferation just goes crazy, right? This proliferation of spines 24 hours later, the day after ovulation, you can see this retraction. Now, one limitation of these experiments is that because they're based on histology, right, they're cross-sectional. So she had to sack the animal at each time point, and these are like camera lucida drawings, so they're not looking at changes over time, and no fault of their own. That was just the limitation of the science at the time. But we now have better tools. So right now, my lab is collaborating with Michael Gord's group at UCSB because they've developed super cool techniques for high-resolution imaging of hippocampal neurons using two-photon microscopy in awake animals. So they use this implanted microprism. It's kind of like a submarine periscope flipped upside down, and they can image the hippocampal circuit at cellular resolution. And in a series of experiments led by graduate student Nora Wolcott, female mice are being imaged every 12 hours for 8 days, right? So across 2 complete estrus cycles. And at each imaging session, Nora's performing vaginal cytology so she can stage the mice, right? Where are they in the estrus cycle? This was all done blind. She actually produced a machine learning classifier that we call EstrusNet. So anybody who's interested in getting into animal work or who works with animals but doesn't know how to stage, Google it. You can find it online. And what she's doing is, so with longitudinal 2P imaging, she's able to track spines on apical dendrites of CA1 neurons over several weeks. And consistent with Woolley's classic result, what Nora found is increases in spine density during proestrus, right here, and then increased pruning during estrus. So overall, there's about a 15 to 20% change in spine from proestrus to estrus, and you can see the cycle, right? Ebbing and flowing, just perfectly coincident with the ebb and flow of hormones, right? So, and what's cool about this technique is that we can actually identify sort of what spine types they are. Are they stubby, thin, mushroom, filipodium? We can see it's the same segment of dendrite that's being imaged in each imaging section. So we can literally see new spines grow and retract. So pretty clear evidence of modulation of spine density across the cycle. But why is the brain organized in this way? Right? Why are these rhythms happening? That was sort of the next question. This was really just proof of concept because we believed Catherine's work, but now we showed it using this sort of new technique. Now let's get at this sort of why question. So the next step was to see how these spine changes influence the functional properties of hippocampal neurons. So now Nora is imaging calcium activity in transgenic GCamp mice while they navigate in a floating circular chamber. So keep in mind that these are head-fixed critters because we have to image their brains, so they're head-fixed, but we are interested in navigation behaviors. So they can't freely roam, although we're working on a movable scope, but we can let the environment move around them. So you notice that there are these sort of different patterns on the walls. That lets us get really beautiful place cell recordings in the hippocampus, right, in CA1 place fields that we can measure these response properties over time. So as an example, there's 2 different environments, let's call them A and B. So, and they have different visual clues around this circular track. And if we just line up the cell's responses to where the animal is in the track A, you can get these really beautiful place fields that respond to very discrete locations around this track. Then you move to B and you see sort of a scrambling of this effect, right? So these cells are responding very differently in track B. But then if you put the animal back in track A, right, A prime, the place fields sort of line up again, right? So there's this sort of similarity. So you can look at 2 different measures, right? You can look at stability, which is the similarity of the population responses between A and A prime, which is what we're coding here in blue. But you can also look at sort of the remapping of place fields between 2 different environments. So the difference between A and B, which is in orange here. And what you see is that when estrogen peaks in proestrus, you see both the greatest stability and the greatest remapping. Right? So these spine changes that we saw on the previous slide seem to be driving these changes, these functional changes within these CA1 neurons. And now the sort of last step of this is to use CRISPR to suppress particular kinds of estrogen receptors to really try to knock down this effect. And then we'll be done. Okay, what about in humans? Can we see morphological and functional changes across the human menstrual cycle? So the lab had this crazy idea to image a person's brain every 24 hours for 30 days, collecting blood and brain scans and mood scores at each time point. And this might have petered out on me. You got it? Okay, and one editor later wrote that they're like old school NASA test pilots. They put themselves in an MRI scanner and drew their own blood every day for 2 months. And I always make the joke that if there's anybody in the IRB in the audience, please rest assured that we did have a trained, licensed phlebotomist. So the first author and the test subject for this was Laura Pritchett. She was really the mastermind behind all of this. And there we go. Okay, so why did, can we go, oh, was that me? Okay, so why did we try to pull off this crazy study? Well, because cross-sectional studies that image the brain at one time point can't tell us anything about how the brain is changing day to day or week to week. And remember what I told you, that a central feature of the mammalian endocrine system is that hormone secretion varies over time. So during an average human menstrual cycle, you see about like an 8-fold increase in estradiol, which is the major form of estrogen in mammals. It's this teal line here. And you see about an 80-fold change in progesterone, which is this blue wave at the bottom. These are on different scales, you'll notice. And the kinds of cross-sectional designs that are typically used in human brain imaging studies don't capture these dynamic endocrine changes. So we were inspired by dense sampling studies pioneered by folks like Russ Poldrack and the Midnight Scan Club, because that kind of like intensely longitudinal imaging lends itself really beautiful to our problem, which is trying to understand how endocrine rhythms may be influencing the brain. So yeah, the 28andMe project was really designed to understand how these rhythms might change or drive physiological functions, because even though we know this in animals, right, nobody really knew how hormones were shaping the human brain on this time scale. So as I mentioned, the participant had daily blood draws and MRI every 24 hours for 30 consecutive days across the cycle, and then she did it again 1 year later. So the data that I'm going to show you are based on a standard 10-minute resting state scan. We used a 3D foam head case because anybody who's done resting state knows how sort of deleterious motion can be. So I'll just brag for Laura that on average she had less than 130 microns of motion every day. Right? It's great to use your own, yourself as a test subject because you're perfectly still. Cortical parcellations were defined from the standard 400-node Schaefer atlas with 15 subcortical nodes from the Harvard-Oxford atlas. And to start off with, we just wanted to test the basic hypothesis that whole brain resting state functional connectivity would be associated with intrinsic fluctuations in sex hormones in just a time synchronous, right, sort of day-by-day fashion. And sure enough, the first thing we observed is that these endocrine changes impact widespread patterns of connectivity in the human brain. Right? So increases in estradiol were associated with greater functional connectivity. Here, oh. There we go. So here, hotter colors on the top are just indicating increased coherence with higher concentrations of estradiol. Cooler colors indicate the reverse. So you can see that estradiol was largely associated with this sort of increased in coherence, and progesterone had the opposite effect, right? Which is really cool to me because in the animal literature, this is exactly what you see, that estradiol has these sort of proliferative effects, and progesterone sometimes can have this sort of opposite. Obviously, we're looking at this in very different timescales, so I won't read tea leaves. And then we can look at this, if we just parcelate the brain into these sort of canonical networks, we can look at this associations across different networks. So you can see that these are evident pretty much throughout the brain, but maybe there's some networks that are more sensitive than others. But this is a time series, so we can use time lag methods to, borrowed from dynamical systems analysis to really test the temporal directionality of these associations. So I won't go through the VAR model, but we're basically trying to solve the question of, is it estradiol that's driving changes in brain states, or is it the other way around? And overwhelmingly, it's estradiol driving changes in functional coherence. And you can see, in particular, it's these regions of association cortex, like the default mode, the control, and the dorsal attention network, that seems to be the most sensitive to these ebbs and flows of estradiol over time. We were in the middle of the pandemic, so we just kept digging through the data. So now we used common graph theory metrics. So for example, to look at sort of the efficiency of any given network, or how well integrated are the nodes of a network. And we found that estradiol enhances within network integration, so global efficiency, in several large-scale brain networks, but the strongest effects were very clearly in the default mode network and the dorsal attention networks. And these data replicated in a follow-up study conducted 1 year later. We kept going, so we teamed up with a group in physics to apply methods from complex systems analysis, dynamic community detection, to provide some of the first evidence that fluctuations in ovarian hormones over the cycle might actually drive the reorganization of the brain's functional connectome. So we see, this is work led by Josh Mueller and Jean Carlson. So we see evidence of greater flexibility of brain networks during this ovulatory window. You can again see it as a time series. Let's see if that can, there we go. And again, sort of ovulatory window being coincident with those peak levels of estradiol, right? So we're able to capture the brain's response to these rhythmic endocrine changes in a bunch of different ways. And I like to include this slide for the men in the room, where intrinsic fluctuations in sex hormones are associated with whole brain coherence in both sexes. So even though I am obsessed with women's brain health, I do not want you to walk away from this talk thinking that hormones are only something that women should consider, right? We know that these dynamic processes are occurring in both sexes, but maybe on unique timescales. So we ran a follow-up study of a densely sampled male. But this time the participant underwent brain imaging and venipuncture every 12 to 24 hours for 30 days so that we could target the diurnal changes in steroid hormones that occur. So during this AM to PM diurnal cycle, we see about a 60% change in testosterone and about a 40% change in estradiol. And just like before, we can see that these increases in estradiol and in testosterone over time are associated with whole brain functional coherence in the male brain. So as Rebecca Shansky asks us, are hormones a female problem? No, hormones are not a problem, and they're certainly not something that's unique to women. So don't buy into that if you see that in the popular press. Couldn't be further from the truth. These are critical neuromodulators for both sexes. Okay. Next, we shifted gears to look at brain morphology. Can we see dynamic changes in brain volume across the cycle? This is an analysis led by Caitlin Taylor. We used a higher resolution T2 scan to assess the volume of different subregions of the hippocampus across the menstrual cycle. Again, every 24 hours for 30 days. And then we reran the experiment, but this time the participant was on a drug that chronically suppressed progesterone levels and selectively suppressed progesterone levels by 97%. So notice this blue wave, this progesterone wave during the natural cycle is absent in the follow-up study. And what did we find? So across the endogenous menstrual cycle, we see these intrinsic fluctuations in progesterone are associated with volumetric changes in several regions. Here I'm just showing two. So a region of the hippocampus called the CA23, and then the outer parahippocampal cortex. And when progesterone concentrations are elevated, right, in the luteal phase, you see this sort of expansion of gray matter volume. And then when you chronically suppress progesterone pharmacologically, you abolish this effect. And here's just the same data showed in a slightly different way. So all in all, I think these results are suggesting to us that progesterone may have the ability to really rapidly and dynamically shape aspects of hippocampal morphology. Okay, in that 28andMe series of projects, the participant took a drug I mentioned that selectively suppressed progesterone levels, sort of like flipping the light switch on progesterone. What happens if we trip the entire circuit breaker? So that was a study that launched 2 years ago. It's run by 2 graduate students, Elle Murata and Hannah Grotzinger, and we're asking that question. So here's a little neuroanatomy for you. Remember I mentioned that the brain controls the release of sex steroid hormones. And it does so because of the activity of just a few thousand neurons deep in a region of your brain called the hypothalamus. These are gonadotropin-releasing hormone, or GnRH neurons, and we can target those GnRH neurons pharmacologically to suppress the hormone production for days, weeks, months, even years. Now, I realize this might sound a little sci-fi, but what you have to understand is that the drug that we're using is an FDA-approved treatment for endometriosis. So half the women in our sample end up taking this drug because they're prescribed it by their doctor. They typically stay on it for 6 months to a year. And what we're capturing is what happens in the brain in response to this form of chronic hormone suppression. So we enroll women before treatment and then about 6 months into treatment. And they're doing this as a treatment, right, to sort of tamp down the immense pain due to chronic endometriosis. But we're interested in what's going on up north, right? How is the brain responding? So we're lucky in that this brain project is funding one of the largest imaging studies of the impact of endocrine manipulation on the human brain, with 400 MRI sessions, expansive fluid biomarker panel, neuropsychological tests, immersive virtual reality measures of spatial navigation. We do multimodal brain imaging and clinical assessments. It is a big undertaking, but our goal is that this project will make major contributions to just our basic understanding of hormone action in the human brain and provide clinical utility for the millions of women around the world who are undergoing this kind of pharmacological treatment in their lifetime. Okay, so we talked a little bit about the menstrual cycle, but let's talk about other neuroendocrine transition states, because the endocrine changes that we see across the cycle are dwarfed by the changes that we observe over pregnancy. So what's going on in the maternal brain? Let's see if I can get that. There we go. So in a recent study that is ongoing right now in the lab, we're following women starting preconception with brain imaging and serological evaluations every 2 weeks throughout the complete gestational window. We've got follow-up scans that go up to 2 years postpartum. And you can see that during the gestational window, right, there's this steep rise in the production of estrogen and progesterone, and then at birth, there's this precipitous drop in hormone production. These are sort of canonical changes associated with the gestational window. And across pregnancy, we can see changes in cortical thickness measures. Again, here we're looking at it sort of the network level, but we can see these network-specific changes, right? Some appear to be expanding, some are maybe getting sort of more fine-tuned. We see this reduction in volume. And you can see this as a function of changes sort of week by week across the gestational window. These are tightly coupled to the endocrine changes that are occurring during this window. So I hope you're beginning to get a sense of how responsive the brain may be to these major endocrine transitions. And here we can look at it in just a slightly different way. So here we're just looking at it by time bin, right? So pre-conception, first trimester, second trimester, third trimester. This is looking within the default mode network, measures of both cortical thickness and gray matter volume. So you can see this sort of stair step, right? You're seeing this increase in cortical thickness and gray matter volume, and these may be permanent, right? Or at least very long-lasting changes, because we can see in the postpartum period, which includes scans up to 2 years out, that these changes are still evident. If we use the higher resolution imaging of the hippocampus, we can see remodeling happening here too, and we can see pretty striking changes within the diffusion, so looking at white matter microstructure. So really quite sweeping changes, suggesting that this period of matrescence, which is what we're sort of now referring it to as, is kind of like adolescence in the sense that it's this protracted period of neuroplasticity in the adult brain. Okay, and what about later in life? What about in midlife, when hormone production starts to decline, right? This is during the perimenopausal period or the transition to menopause. You can see that over a fairly short time window, right, production of these sex steroid hormones begins to change. So again, let's look at, I'm always inspired by the animal literature. I tend to go there first to see what they've figured it out. So here's work from a non-human primate model comparing, again, those dendritic spines. This time we're in the prefrontal cortex. And we can see that surgical menopause seems to induce this sort of retraction of spines within pyramidal neurons in the prefrontal cortex, and that an estradiol add-back regime restores spines and preserves memory function on a delayed response task. Okay, what about in humans? Well, the menopausal transition, as I showed before, we know results in this sort of substantial rise in gonadotropin, so a hormone called follicle-stimulating hormone, or FSH, and a decline in sex hormone production. And during this period, about 40% of women will report changes in mood and attention. Sometimes this is labeled as menopause fog. And yet, the influence of endocrine aging on the human brain remains severely understudied. And this is kind of a quantification of this knowledge gap on the right. So over the past 30 years, the neuroimaging community has done a pretty impressive job of trying to understand and try to map the brain as a function of chronological aging, right? Using this sort of canonical model of looking at men and women who are 65 and older and comparing their brains to young adults. Like, we've done a great job of understanding neuroanatomical changes, neurochemical changes. But what about this uncharted decade in the middle, right? 45 to 55, 45 to 60-year-old window. This coincides with menopause. And you can see that the number of publications on endocrine aging or menopause are just totally dwarfed by the number of studies using that sort of canonical aging approach. So there's a lot of work for us to do to understand how endocrine aging might shape the brain. And I hope I've convinced you so far that this is a pretty worthy question to ask. So right now, my lab is studying the extent to which endocrine aging shapes all aspects of the brain, but really sort of zooming into 3 different levels. So at the mesoscopic scale, just whole brain intrinsic network organization. At the submillimeter scale, looking at the kind of higher resolution hippocampal subfield morphology. And then at the circuit level to understand whether disruptions in the mechanisms of learning and memory may contribute to these symptoms of menopause fog. And really to understand those amazing women who just seem to like sail through menopause with nary a worry. We really want to understand their brains. So just to show some new data that maybe I didn't get into yesterday or in discussions, one of the things that we're really interested in is spatial navigation. So we use fully immersive ambulatory assessments of navigation ability to look at different aspects of navigation behavior. Here I'm just showing 3. We actually have like 8 different assessments. But we can look at things like path integration or navigational strategy. So let me just walk you through one of these examples. So in this maze task on the far left, task number 3, it's called the dual solutions task. So subjects put on these goggles, they're in this big open arena, and they're navigating through this maze, this virtual maze. And you can follow this purple line here. So they're actually led in a training phase. They're led around this maze. Follow this purple line. And those black diamonds represent objects that they'll encounter along the way. And then in the actual test phase, we plunk them down randomly at one of these objects and we say, go from object A to B, or C to D. And we just look at the navigational strategies that these individuals take. Do they follow the learned route around the periphery? Do they take shortcuts? Or are they like me and they just wander aimlessly around? And we can see that there are differences with aging. So these heat maps just represent the kind of propensity maps of where these people are walking. So young males seem to plow straight through the middle of the maze, while we can see that in midlife males and females, they're kind of hugging the periphery a little bit more. But what's going on around the time of menopause, right? We can see these interesting changes so that around the time of perimenopause extending into postmenopause, there's sort of this increased preference for doing those learned routes around the periphery, so less likely to take shortcuts. Now note that there is no difference in their ability to get to the target location. So their accuracy is spot on. They can perform the task just as well as anybody else. They're just using different strategies to get there. And what do we see under the hood? So from preliminary data, we see emerging evidence, just like we saw in response to hormonal birth control or pregnancy, that endocrine aging may influence medial temporal lobe tissues. So during this transition to menopause, again, you see two things happen. You see this decline in estradiol and progesterone production and this increase in FSH, follicle-stimulating hormone. And what we see in, if you look at postmenopausal women, that variance in this FSH signal. So postmenopausal women who have higher levels of follicle-stimulating hormone are those that also show sort of the lower volume within many of these medial temporal lobe structures. So hippocampal CA1, CA2-3, entorhinal cortex, and subiculum. This is preliminary data. We haven't worked our way through the whole sample yet, but this is the signal that we're seeing so far. And our findings linking FSH and medial temporal lobe volume in humans comes on the heels of a recent report in mice suggesting that FSH may act directly on hippocampal neurons and cortical neurons to accelerate amyloid beta and tau deposition and impair cognition, and that blocking FSH stalls these effects. Okay, so a few things to sum up. So in keeping with the animal studies, our findings suggest that these HPG axis hormones, hypothalamic pituitary gonadal axis hormones, shape the structural and functional architecture of the human brain, maybe particularly in memory circuitry. That early alterations in memory circuitry are evident decades before the age range typically targeted by cognitive neuroscience of aging studies. Circuits that may be most susceptible to aging and neurodegeneration, like the default mode and the medial temporal lobe tissues, are densely populated with estrogen and progesterone receptors and modulated by sex hormones. And so I hope I've convinced you that a complete understanding of the brain really must include a role for sex hormones. Okay, so I've presented some data using sort of precision imaging approaches where we can scan small numbers of women but do so over like really rapid fire timescales and kind of really focused, so small n, but deeply characterizing the brain over these neuroendocrine transition states. But as former DESPO lab member and good friend Katerina Grattan writes in her sort of opinion piece of like, what's the future of cognitive neuroscience, she argues that it's sort of one of these two directions, right? We have to go big or go small. And so I've shown you our sort of precision imaging approach, but what can we do at the consortium level to ask some of these questions? And this is our effort to do this. So we just a few months ago kind of secured funding to launch the Ann Bowers Women's Brain Health Initiative. And the idea is to take women's health prime time. And at the University of California system, and maybe with strategic partners around the country, right, we can do that through deeply collaborative science. So across the University of California, we've got 8 brain imaging centers dedicated for research, and these generate data from thousands of individuals annually. So we're just capitalizing on that activity to create a population-level, open-access brain imaging database that just pools brain imaging data across these centers and then pairs it with de-identified health information. And it's really sort of unabashedly designed to serve women's brain health. So wish us luck. We're just starting out on this endeavor, but we're excited to see where it goes. And with that, I'll just say thank you for your attention. And if you're ever in Santa Barbara, come say hi. The view's not bad. And a big thank you to all of the folks who actually did this hard work and produced all of this science. And thank you for your attention. Happy to take questions.
Okay, Emily, thanks so much. Incredibly eye-opening talk. Lots and lots of things to consider here. Let's go ahead and kick things off with questions. Just raise a hand if you have one, and we'll send a mic around to you.
Why does classic aura with migraine affect options for birth control?
So migraines are, there's major sex differences in the presentations of migraines, and there's an emerging literature to suggest that they're quite sensitive to fluctuations, in hormones. So sometimes the pill is going to be given to try to mitigate migraines. For some women, it has the exact opposite effect. So I would say that the science is still, quite behind in how we can think about the role of steroid hormones in migraine use, and especially, different subtypes like those with or without aura. But there is a literature, and there's actually it's funny that you should ask this, because one of my very first honors students, her name's Morgan Fitzgerald, she suffers from chronic migraines, has not, pain scale of 7 out of 10 every day, has been at Mayo to do clinical trial. She's exhausted every resource and hasn't found much that works for her. And she's now at UCSD getting a PhD to ask exactly this question. So look up Morgan, because I have a feeling in about 5 to 10 years, she's going to be the one to answer that question. Takes living through the experience to drive the scientific question sometimes. Yeah.
What does the DMN refer to in that slide?
What is the DMN? So it's the default mode network. So this is what used to be called this sort of task. So when you're performing a task, right, you see the default mode network activity go down. But when you're just sitting in the scanner, your mind's at rest, you're sort of ruminating about past events, you can see the default mode network activity increase. So there's lots of different subdivisions, right? We used to think the default mode was sort of like one big network. It turns out maybe there's lots of different subdivisions in it. And we're beginning to see effects of steroid hormones on some of those subdivisions in particular. But it's,
You mentioned briefly at the beginning that we know virtually nothing about hormonal birth control. What do we know about it? And I know you had mentioned things about dopamine and what are some things we do know?
Sometimes I add slide. No, I don't have these in my slide deck. But yeah, Mark and I just published a paper last week. I think it got accepted. Asking this question in a somewhat basic manner. So we were looking at women who had gone through a molecular PET imaging study looking at sort of 3 different aspects of the dopamine system. So dopamine synthesis, stimulated dopamine release, and dopamine binding. And we just, my postdoc was really interested in this question of how, on what scale might oral contraceptives be acting? So we can see it in terms of morphology and function, but what about neurochemical systems? Because again, we know that there's this link from the animal literature with sex steroid hormones maybe being able to modulate aspects of the dopamine system. So she combed through this wonderful data set that Mark had that looked in men and women on these 3 different aspects. And she categorized the women based on whether they were naturally cycling or users of hormonal contraception. And what she's found is this, across the board, hormonal contraceptive users had increased dopamine synthesis, no changes in the other aspects of dopamine function, but greater dopamine synthesis capacity. And also tied to that, they had increased cognitive flexibility on this task switching. So, and from previous literature, we know that those two seem to be yoked, that synthesis capacity might tell us something about this task switching. And sure enough, that's what we find. So yet again, it sort of adds this another layer of complexity of sort of how these major sort of hormone modifications might be impacting the brain at all of these different levels, including basic properties of the neuromodulatory system. But we need to now, I think that's really inspired us to follow that up and design an actual study where we can enroll women maybe prospectively and look to see whether we see the same things when we sort of control for maybe the type of hormonal contraceptive. So as with all good science, I feel like one finding is just sort of opening a door to doing 10 more studies. But
I wanted to ask a default mode network question. So
yeah.
Assuming that there's not more estrogen receptors in the default mode network, how do you think estrogen can specifically influence one network, or do you think it's something else that's just correlating with the fluctuation with estrogen? Or how do you think that works?
Yeah, I mean, what's interesting is that it's not ever specific to default mode, although that's the one that seems to be popping up time and time again, but we see it within dorsal attention as well. I don't know if this is, one thing that I think we really need to do is just look at HRF changes and see whether, how sex hormones are just affecting basic properties of HRF, thinking about regions that might be more or less vascularized, and thus steroid hormones, which we know can have, affect vasodilation and vasoconstriction. Like maybe it's just as simple as that. Yeah.
The estrogen receptors in the brain is not regional.
Well, first of all, that's a great question, Mark. So here's my answer. Our knowledge is based on predominantly non-human primate maps of receptor expression. There's been, so of the Allen Human Brain Atlas, so this is, we've got 6 postmortem brains that have now been stained for various proteins and tissues, and from that, we can look at where these receptors are. But of those 6 postmortem brains, one of them is a woman. And by the way, she's a postmenopausal woman. So, we literally don't even have good maps in the human brain of where these estrogen receptors are. We are relying on 20-year-old data from a different species. So I can't even answer a basic question like that, right? So we know that John Morrison has gone in and done sort of quantifications of estrogen receptors within prefrontal cortex, but maybe that's just a spotlight of attention effect. Like maybe there's receptors other places. Maybe there is this overlap between default mode network and receptor expression, but we just don't have those data. I think that might be coming. There's, I think, just released a new PET ligand that might be able to tag on to ER and PR receptors, which would be huge. And we could then begin to answer that questions really sort of across the lifespan. Like, where are these receptors? How do they ebb and flow over different neuroendocrine transition states? And do they map onto these other ways in which we think about the brain, like these kind of intrinsic networks? So stay tuned.
Emily, I'll ask a quick follow-up sort of related to that. It seemed like in many of your studies you're collecting a set of biomarkers and other demographic and neuropsychological tests in addition to the looking at the maybe brain network to hormone coupling. I wonder if you could give a sense if hormonal levels are changing and say network levels are altering. Is there anything in cognition that, or what is the most correlated element of cognition that you're measuring that would give us a sense of what that means for kind of like daily life function?
Yeah, so, across the menopausal transition, the thing that seems to be the most consistent finding are these changes, and I don't have this slide, I showed it yesterday, but these changes in verbal episodic memories, verbal memories across the board, but really sort of episodic. So things like face name associative memory tasks or list learning tasks, where we can see, a small but consistent decline. So pre and peri are sort of performing high, and then you see this decline in postmenopause. But we always then sort of show the benchmark data of like age-matched men, and you can see that women sort of have this advantage throughout life, and that decline in postmenopause is really just like a decline to the level of men, which I think is important so women don't sort of catastrophize. We don't see consistent changes or really any changes in working memory, selective attention, so more sort of executive function type tasks. This really does seem to be something specific to the episodic memory domain.
Great talk. I'm not a neuroscientist, I'm an engineer. So we're always looking at, I guess, providing technology, collaboration, and research. I think we may have something that that can be useful. You mentioned about the current mice have to be, I guess, fixated at the head and then move on the rotating platform. We probably can provide something almost like the VR ambulatory type environment in an open environment, even with a constrained maze-like structure, and we can process the image and project whatever is needed for the mouse to navigate through. So that
would be great.
Yeah, some technology that we can provide.
I'll get your information, and we should talk more. So this is led by my partner in crime, my husband's lab, Michael Gord. But they have used both the head-fixed physical mazes, and they have done open field VR mazes. So they've got the animals doing VR, but if maybe you are even further ahead on the game, so it would be great to put our heads together and see. They're even thinking about kind of flexible, movable scopes. They're working with a company actually out in California to see if we can truly let them have free ambulatory range with just a scope that is flexibly moving with them. So if you have any thoughts on that, that would be great too.
Yeah. Our technology, I think everyone here has already enjoyed. You probably don't know about it. Have you heard of DLP, digital light processing?
Okay. Yes.
Okay.
Yes. This is
it's
you.
All, yeah. I mean, 95% of the theater in the U.S. and North America are using DLP. So, anything imaging, projection. That's our expertise.
Okay. Well, then you are the person we need to talk to. Thank you for that.
So Emily, very outstanding talk again, really brilliant. What do you wish doctors knew that would help guide women? If you could just say, with the limited amount of knowledge that we have, and you don't want your doctor to just say, oh, well, we don't know here, just try this. What do you wish they would say? And who? What doctors?
My wish list is so long for Santa. Yeah, I mean, as I mentioned yesterday, we need more training, right? So as I said, 80% of medical schools don't even have a basic module on menopause in the curriculum. So, and for those that do, they're mostly an elective. So I think we have to train doctors on the latest science that is available while we push forward that science even more. But there's so many cases. I mean, let's take endometriosis. I mentioned that we're doing this big endometriosis study where we follow patients who go on this drug. But endo affects 1 in 10 women, and the timeline between first symptoms and a successful diagnosis is on average 10 years. Women go a decade, right, in excruciating pain, undiagnosed, because doctors don't know what to do about it. And this is personal, right? It hits home. My grandmother had endometriosis, didn't know it at the time, but once it finally got diagnosed, she was 27, and the doctors gave her a complete hysterectomy because they literally didn't know what else to do other than to just cut it out. We can do better, right? I am confident that we can do better. We need new R&D to think about tissue-specific drugs that don't just affect the entire hormonal system, but maybe can affect tissue-specific receptors. So I think we just have to get physicians, scientists, and physician scientists as a pair to move the needle on all of these fronts. Endometriosis, PCOS, the birth control pill, like all of these suboptimal, important and critical, but suboptimal treatments that we could be 20 years ahead on if we had just taken them seriously from the get-go. And I don't know, maybe I'll end with this. He did include this slide, let's see. This is a book I quite enjoy. This is by a historian, Eleanor Cleghorn, British historian, and she published this just last year. And it really makes the case that medicine has been fearful of women's bodies throughout history. Right? This dates back to the time of Hippocrates, who proposed that a woman's womb could wander around her body. So if she had a headache, it was because her womb was just like hanging out up here, right? It can wreak havoc. And so Eleanor is really kind of just chronicling how how medicine has failed women by treating our bodies as kind of alien, right? As inferior, unknowable. Like literally, the Greek root for uterus means hysteria, or for estrus, it means frenzy or madness. So Cleghorn writes, medicine has insisted on pathologizing femaleness, and by extension, womanhood. But our diseases are not elusive to us, right? But something about our diseases seems to thwart and frustrate medicine at every turn. This dates back to the 1800s, right? The time of Charcot, the Salpêtrière, who handed out hysteria sort of as a diagnosis for any female pathology that physicians just failed to understand. And I really like what she says here, which is that our lives depend on medicine learning to listen. These are not new problems, they're quite old, but we can do better and we will.
Okay, Emily, thank you so much. Let's thank Emily Jacobs once again. Many big issues that we could have gone on much longer. We did not get to online questions. I apologize for that. But Dr. Jacobs is easy to find online, so reach out to her if you had something you really wanted to ask about. Thank you all for being here. We'll be back next week with Anila DeMello, who's going to tell us about the cerebellum and cognition development disorders. So see you then.